Hi, everybody. I'm sorry I can't be there in person. I am currently dialing in from a hotel in Prague where I am. I went to a conference, like a ceremony for the opening of a new lab where I'm going to be working. And hopefully this talk will explain a little bit more about what's going on there. But yeah, basically this is kind of like a last minute thing. It's so please excuse me if it's a little bit haphazard. I'm doing my best to kind of just record this and send it to you today in time. Um, so basically, I'm going to be talking to you about research I've been doing in Croatia. I'll make this work for some reason. There you go. So basically, research I've been doing over the last few years in Croatia, and essentially about what makes it an advantage or a disadvantage to use a standard technique for doing surveys for bats. So if you have any questions, then you can get in touch with me. These are my details. Just drop me an email or just hit me up on socials, and or you can just message Becky. I'm sure she'll pass on messages. But basically, I'm currently in the process of moving to the Czech Republic, to the Czech Academy of Sciences, for this new lab. But I also still do research back in the UK, in Birmingham, Black Country, with the Bat Group, with the Urban Bat Project, and in cooperation with the University of Wolverhampton. And also, I've been volunteering as a bat scientist for Operation Wallacea, which is kind of a biodiversity charity, a conservation charity, since 2021. So I will explain as we go. But just to kind of like tell you about the site that I'm going to be talking about, Krka National Park is one of 8 national parks, designated national parks in Croatia. And there's quite a great deal of protected space, but 8 of them are national parks, and Krka is one of them. It's situated around the Krka River Gorge. And if any of you know this part of sort of like south central Croatia, so Croatia is sort of shaped like a horseshoe, and it's on the Mediterranean side, slightly inland up on a plateau, and the site is a river gorge, and you may associate this with waterfalls because that's the big tourist attraction for the area. But basically, the same sort of geological and hydrological processes that create these incredible waterfalls are also what make it such a biodiversity hotspot. So we are based, the camp is based where you can see the star on the map at the northern end of the park. And we do most of our surveys in and around the gorge, and you can see the border of the boundary of the national park is hatched in green there, but some of the work we do is sort of maybe within a kilometer or 2 of the boundary. So this is what the habitat looks like. So this is a picture looking down the river gorge, and so it's basically like a dry Mediterranean scrub plateau that surrounds this gorgeous river gorge with beautiful water. And the habitat is fairly homogeneous. It's like a juniper and oak scrub, and it's very dry. The landscape is karst, which is basically a limestone landscape through which we get very, very little surface water. So when it rains, the water percolates straight down, and it carves out these amazing gorges, waterfalls, caves, features. But it does mean that there's not a lot of surface water, like ponds or streams. The main water in the area is in the river gorge itself. It is the Krka River. So this landscape supports a number of different mammals, some really charismatic mammal species. And I know that I was supposed to have changed this slide because apparently it's a beech marten. I mean, it's a pine marten. I was supposed to put a picture of a beech marten, but hey ho, sorry, Jenna, who told me off. But basically, we get a lot of these really charismatic mammal fauna: wildcats, jackals, beech martens, and wolves. I've yet to see a wolf. I have heard them on bat surveys there, which is a little disconcerting. But those of you who have been out on surveys at Dudley Zoo know what it's like to hear a lion roar while you're trapping. So imagine what it's like with wolves, not in cages. But basically, we have 25 terrestrial mammal species in the park. The ones that you can see in red here are ones that we haven't actually recorded, but we're pretty sure are there. So we've just submitted like a new paper that is like a new checklist for mammals of Krka National Park paper. So hopefully that will be published soon. But yeah, of these terrestrial species, I mean, a lot of them are hard to record, and we don't necessarily have recent verified records for. And that's just to do with the idiosyncrasy of mammal ecology. A lot of them are very elusive, a lot of them are nocturnal, and a lot of things are recorded basically based on evidence of their presence rather than like actual sightings or actually having the animals in the hand. But as Carl Sagan says, evidence of absence is not absence of evidence. And so but we have no reason to believe that these unrecorded species are not actually there because they're persistent in the surrounding landscape. So, but this only represents half of the mammal fauna of Kirker National Park. The other half is bats. So I'm really hoping that the videos on this talk work and they record okay. This is a little horseshoe bat. It's a Mediterranean horseshoe bat, I believe. I think. Yeah, Mediterranean horseshoe bat. So the park supports 24 bat species. The ones that you can see, I've been written near threatened or vulnerable. These are their Mediterranean or European assessments. And then you can see if their populations are declining, there's a red downward arrow. If they're going up, it's a green arrow up. And then the yellow are stable and quite a number of question marks where we don't know how well they're doing. The two species that you can see in the bottom, the party-colored bat and European free-tailed bat, we think are there, anecdotal records and some acoustic records from the area, but we haven't had those in the hand, but the other ones we have. So basically, what we're looking at is about 42% of the bats in Kirka are of conservation concern. So, near threatened, vulnerable, or data deficient. And that number tends to be kind of the magic number for bat conservation status worldwide. So about 40 to 50% of bats worldwide are of conservation concern, the same percent in Europe, the same percent in the UK, and the same percent in the Mediterranean. And the same is the case for Kirker National Park. So, approaching 50% of bats are in real trouble. So work in Kirker National Park started in 2018 with just some tentative surveys. And so I'm going to tell you now kind of the story of how the bat science has evolved there. An interesting thing about the bats in Kirka is that because of the nature of the habitat being scrub, there's very little in the way of mature vegetation. So most of the bats that are there tend to be those that roost in trees, not in trees. So there'll be building roosting bats, cave roosting bats, and even as you can see in the top right here, that's a Bechstein's maternity colony that is actually in the field study center where our base camp. They come back there every year and roost in the roof, which those of you who are familiar with Bechstein's bats are strongly associated with woodland, but not in this case. So they have quite a lot of roost fidelity. I would really like to start ringing these bats. We'll see what happens. But you can see in the video here, there are two lesser horseshoe females, and you can see they've got their babies as well. And they are in buildings very similar to the one in the middle on the right. So there's a lot of abandoned farm buildings. This is an abandoned school building. And they're a really vital resource. They've become a real vital resource for Rhinolophus maternity colonies in particular. And so we do a lot of revisiting ones that we found to see that they're still in use. So, and then there is a number of mines and tunnels that are human-made, but by far the most important resource for bats in the area are the caves. So as I was saying, the landscape lends itself geologically and hydrologically to creating these amazing karst caverns. This site that you can see pictures of here is Miljatska 2, and it supports 9,000 bats of at least 8 different species. And I'm just going to show you a little video, which is just in the very entrance, just to give you an idea of the kind of density of bats. Most of the bats that you can see in this video are Myotis capucini, long-fingered bat. So of the 9,000 bats in this cave, 7,000 of them are Myotis capucini, and you'll also probably see a few horseshoe bats hanging around in there. But a lot of cool bats, many bats that we have in the UK, but also things like greater and lesser mouse-eared bats, and Geoffroy's bat as well. And 4 or 5 different species of horseshoes, probably 4 recorded so far. So one of the things that we do is, so the way that OPWAL works, Operation Wallacea, is that it's a student-funded conservation and research model. So students pay to come and have an experience in the field. But rather than it being sort of like a very sort of like ecotourism based model, they're actually coming to take part in ongoing research. So they really get stuck in, they get lecture series. Some of them will stay for a week, some of them will stay for the summer. So it depends on their age, whether they're a school group or whether they come, you know, a gap year kind of thing, or as a research assistant or a dissertation student, which we get a lot of those. And then the science team tend to be like people who are early career conservationists, usually, not always, but usually people who have just finished a master's, say, that kind of thing. And they get to have the experience of doing field work for the whole summer and living in the camp and learning things like, teaching, which is something that a lot of master's students don't necessarily have. So it's a really kind of good upskilling for everybody. And they do get to see some animals. You can see here there's a bat being weighed and another one being processed on the bottom right. But the key thing for the bat team is taking students out to see what it's like doing a mist netting survey, like a nighttime mist netting survey. But we have a slight problem. So this is a photo of 16 of the 24 bats that we have, 24 bat species in Kirker National Park. But it's also the number of individual bats that was caught in the first 3 years of mist netting on transects. So 3 years, 3 seasons of surveying for 6 to 8 hours a night for 5 nights a week, and this is how many bats we caught in 3 years. So something was kind of going wrong, and we needed to kind of look to see if there was something that we were doing wrong, or that we could tweak to kind of make the surveys work better. So to put this into context, we talk about the transect model next. So basically, transects are selected when they're setting up a site and they are to reflect changes in habitat. So the different types of scrub, the different and like the different topography of an area as well. And so the transects take place, and lots of different surveys take place on transects. So like mammal camera trapping, herpetology, butterfly transects, songbird counts, but usually mist netting for bats as well. But this is what we were doing. We're basically going up, putting up 45 meters of mist net and trapping for 8 hours, and most of the time catching nothing. So we didn't really know what was going wrong. And if I just show you, for example, the difference between the Galapagos, Mexico site and Kirker National Park, these pictures are at the same scale. So obviously there are some differences. We try to control for those in our data analysis. So we look at our survey effort in terms of net hours. So you can compare a site that you can only fit short nets to a site where you can fit really long nets by saying how many meters of net did you have up per hour. So 12 meters of net per hour is a net hour. And that enables us to be able to compare between sites. So what we looked at was the number of bats caught per net hour and the number of species caught per net hour. And also because we know that the Mexican site has a much higher species assemblage, much larger species assemblage, we looked at the percentage of the known assemblage caught per net hour as well. So that enabled us to go. So you couldn't just go, oh, well, of course they catch more in Mexico. They've got more bat species in Mexico. So that's corrected for. And you can still see in the box plots on the right-hand side that the catch efficacy at Kirker National Park, the ones that say K and P, is much lower than at the other sites, using exactly the same method. And the same correcting for the same survey effort. And these types of surveys work so well for invertebrates. They work so well for snakes, for songbird counts, for mammals, all kinds of stuff. Why weren't they working for bats? What was going wrong? And the key all comes back down to that habitat that I talked to you about. The fact that there is very homogeneous landscape. Everything is very much the same, and it's scrubby, but there's no woodland edge. There are no streams, very few rivers, and very few standing water bodies. So no ponds, And basically, the transects were selected because of the different types of scrub habitat, and they were put up. But there was no linear features along which bats would normally commute. So bats would normally go from their roosting place directly to their foraging grounds, in the most efficient way possible, and in the way that gives them the most sense of reducing perceived predation risk. So nice cover. But there is no cover. And so basically, essentially putting up bat nets on transects in Kirka was basically like a needle in a haystack stuff. You're putting up nets and just hoping a bat flies into it. So we decided to kind of tweak things slightly. On our nights off in 2021, we were frustrated with the fact that we weren't catching anything on transects, and we decided to start doing some additional surveys on our nights off. And we would go and we would put 3 meters, maybe 6 meters of net up somewhere, but we put them up in targeted areas. So areas where we knew or expected bats to be present. So this area is called Castagna Pond that you can see a picture of here. And that's where we first trialed out the Skynet. So this is the Skynet. This is Ollie, who designed the Skynet. He's a herpetologist and a proponent of what he calls ropey science. And Ollie designed this net to be suspended from a rope over the middle of a pool. And we've used this design now to great effect in several locations back in the UK as well. We did this as part of the Urban Bat Project. And it really made a difference. Just going somewhere where we knew where the bats would be and putting nets in the flight line, in the anticipated flight line. This is how the Skynet works. It's kind of a cool video. It just gets pulled out into place and then pulled back in, and then it has these little ropes for tension on either side. We did publish this, and there are how-to videos if anybody is a bat trapper and you want to know how to make it. All the information and equipment list is there. This is the paper. It's open source, so you can access this as well. And so basically, how do we do in our nights off in that week, in that season, sorry. So on transects, all season, we caught 4 bats. I think of 2 species, so like natters and possibly a horseshoe bat. And that's all we caught. On our nights off, on the targeted surveys, we caught 42 bats. And bearing in mind, the 42 bats were not caught in 8-hour surveys at night. They were caught in a 2-hour survey at dusk. So that meant that the students could come and be there for the whole survey rather than being out all night with us as well, which made a big difference going forward as we started to bring this into to be our main survey method. So we talked to Opwall about this, and they were incredibly supportive and enabled us to kind of rethink the way that we were approaching the surveys. And we decided to move to a constant effort site model. You might be familiar with a CES model because that's what birdwingers use. Usually they will go to the same site, put nets up year on year in the same locations, and enables them to get usable data over time. So now we trap alongside buildings, we trap outside of tunnels, we trap along trails, and it gives us a lot more flexibility. And we have spent the last few years finding the best sites that we can then start to do long-term monitoring. The other cool stuff is we get to do things like team building activities. So this is one where the students get to design and construct a net to go in the front of this mine to catch bats coming in and out. And this is Paulina, who was supposed to be doing the talk tonight. This is her extracting from that net. So just kind of like we get to actually teach that sort of like fieldwork innovation approach as well, which has been a great benefit. And now we get our feet wet. So this is one of our constant effort sites. It's the Brilliant Dam, and it's really cool, and it's kind of a lot more fun as well to kind of do stuff that feels a little bit more like adventurous and jungly to go out and be able to just sort of like, stand in a lake and put up mist nets and know that it's going to work because going and putting up these nets and knowing we weren't going to catch anything and knowing we were just going to have to sit in the dark for 8 hours can be quite demoralizing, and you end up not getting the scientists coming back and wanting to come for future surveys, and we're now getting better staff retention as well. So this is how it's ended up. So in the 3 years, as I said, we caught 16 bats, and using constant effort sites, we've caught 455 bats in the same space of time. most of which have been in the 3 years since we switched the model. And then also this is supplemented by in-hand sort of hand netting, where we go to, known roosts and checking on those rhinolophus roosts in the abandoned houses as well. So we're doing a lot better and actually getting really good usable data. The disadvantage is it's now not comparable to sites like Mexico because we're not using the same survey method. However, when we weren't catching anything, there's no meaningful data being gathered anyway. So, so we've just published the results of this study. Well, it's in publication at the moment. It's just been accepted. So, if if you follow the back group or you follow me on socials, then you'll get told when this is out. But yeah, we're really excited that quite a lot of years of research have now gone into this, and we feel quite vindicated in our decision to change the survey method. So what we've got going on coming up is we're working with Apodemus to hopefully do some lure trials on our constant effort sites. And if we weren't doing constant effort sites, we wouldn't be able to really do that if we weren't catching well. And the other thing is ongoing work looking at obligate parasite systems. So looking at bat flies, the parasites that live on the bats, and also the fungi that live on the parasites. So this multi-trophic system in which if you have, and there's a lot of host specificity in the system, so you're looking at the potential for greater species loss than you anticipate if the primary host, i.e. the bat, is of conservation concern. So that's all stuff we've got coming up. I just want to say thank you to everyone who has helped over the years at Kirkin National Park, and long may it continue, really. And if you have any questions, like I said, just drop me a line on socials or email me, or just I'm sure that you can ask questions in the online chat here. Really sorry I can't dial in, but very busy setting up for lots of bat stuff to come. So yeah, thanks for having me. Bye. Oh, and happy Halloween.